Finally, the mechanotransduction. This is the process where the deflection of the inner cells in the ear, so the mechanical deflection, is converted into electrical signals that generate in the neurons electrical signals, so action potentials, that are carried away by the cochlear nerve to the auditory cortex. So, for the process of mechanotransduction, we need to know the following. The endocochlear potential. We have here the endolymph. Remember, the endolymph here has a high concentration of potassium, okay? Now, the potential between the endolymph here and the inner hair cell is about minus 70 millivolts. So this is the potential difference between the endolymph and the inner hair cell. Potassium concentration in the endolymph is very high. It is slightly higher than the concentration of potassium inside the inner hair cell. So there's a slight chemical force driving potassium ions into the inner cells. However, there's a much higher electrical force driving potassium into the cell. So we have a force driving potassium ions into the inner cells. The question is, how do potassium ions go into the cell? They go into the cell through these channels, which we see here. These are mechanosensitive potassium channels. The sound wave deflects these hairs. These hairs are known as stereocilia, the shorter ones, and the longest one is generally called the kinocilium. So the deflection of these hairs toward the kinocilium pull on the hair cells. Now, the hair cells, as we can see here, are connected to one another by structures that are known as tipplings, which we will also see in the next slide. So, the mechanical wave, the sound wave, deflects these stereocilia toward the kinocilium. This stretches the tipplings, and the stretched tipplings pull on the mechanosensitive potassium channels, as we see here. These potassium channels then open up. Since we have a driving force for potassium to go into the cell, potassium will go into the cell. Now we have here calcium channels, voltage-gated calcium channels. Potassium, which polarizes the inner cell, opens up the calcium channels, calcium flows into the inner cell, activates the vesicles to release the neurotransmitter glutamate. Now, glutamate binds the receptors in the neurons, which results in action potentials, as we see here. Deflection of the inner cells, as we can see here, generates a higher frequency of action potentials. I would like to stress, we always have some activity going on, as we can see here by this frequency of action potentials. Now, this frequency of action potentials can increase as we deflect these cells. The action potentials that are generated in the neurons upon the deflection of the hair cells, okay, these action potentials that are generated in neurons are collected by the cochlear nerve and carried all the way. Remember, we need several neurons, but I will discuss this in a separate part. So these electrical signals as action potentials are taken by the cochlear nerve all the way to the auditory cortex. Here we see again a summary. These are the cells. They get deflected. Here we have the tipplings. They get also deflected. They get stretched. As we stretch the tipplings, we open up the mechanosensitive potassium channels. Potassium goes into the cell. We have depolarization, opening of calcium channels. Calcium goes into the cell, activates the vesicles to release the neurotransmitter glutamate. So here we have action potentials in the cochlear nerve. Finally, the cochlear nerve carries the signals from the inner hair cells to the auditory cortex. As we see here, we have 7 neurons, and here we have the auditory cortex. I will discuss the details in a separate part. I hope this was helpful.